Faculty of Agriculture. I take immense pleasure to welcome you, sir, to Mangalayatan University. So today's guest is Dr. M. Sheshu Madhav, Director of ICAR Central Tobacco Research Institute, Rajahmundry, Hyderabad, Andhra Pradesh. Sir will deliver his talk on the topic Biotechnology, a promise to farmers, prosperity, and next generation agriculture. Sir, I will take an opportunity to introduce you to the audience. Sir is having 25 years of research experience in the field of plant molecular biology and biotechnology. He has immense experience in molecular genetic tools for breeding of rice, tobacco, and citrus. Now, I will take you to his academic profile. He has completed his bachelor's in agriculture from Andhra Pradesh Agricultural University, Hyderabad. Then, after that, he has completed his master's in agriculture biotechnology from Assam Agriculture University, Jorhat, Assam, India. Then is pursued a doctorate degree in molecular biology and biotechnology from a very renowned institute, that is IARI, Indian Agriculture Research Institute, Delhi. Further, sir has pursued his postdoctoral research at Ohio State University, Columbus, Ohio, United States of America. Sir has served at various institutes at different capacity. As I have already introduced that sir is presently serving as a director of ICAR Central Tobacco Research Institute. Previously, sir has served as principal scientist biotechnology at ICAR Indian Institute of Rice Research at Hyderabad. He has also served as senior scientist at Directorate of Rice Research, Hyderabad. Then scientist at his present institute and NRCP, that is which is at IARI campus. Sir has successfully completed 22 research projects, and he has in national and international publications 178 publications. Now I will take you to his Google Scholar profile. His citation is 3,680, H index of 33, and I-10 index of 94, which is really commendable, sir. Besides this, sir has developed several technologies and several varieties such as VRR, Han 51, 55, 57, 60, and which are all for disease resistance against blast. Sir has also having several awards and honors under his belt. To mention a few, first one is Fellow of National Academy of Sciences, Fellow of Royal Society of Biology, London, UK. He has been also awarded the Lal Bahadur Shastri Young Scientist Award from ICAR, Government of India. Then National Recognition Award in 2020. These are a few to name. The list is really very long. Sir has also served as academic editors for some reputed journals such as PLOS One, BMC, Plant Biology, Annals of Genetics. Besides this, sir has also served as Institute Biosafety Officer at IIRR to look after the biosafety issues. Sir has many national and international collaborations. To name a few, with Dr. G.L. Wang at Ohio State University, with Dr. K.K. Juna at IRR, Korea, and with Dr. T. Mahopatra and Dr. N.K. Singh at IARI. So, sir, it is my immense pleasure to welcome you at Mangalayatan University, and we, the Faculty of Agriculture, as well as our students, are eagerly waiting for your address. Thank you, sir.
Yeah, thank you very much. Honorable VC, sir, can I start now?
You can start, sir, now.
Yeah. So good morning to all. Honorable VC, Dr. Samshivarav Garu, and eminent scientist, Dr. Professor S.K. Rao Garu, and the Faculty of Agriculture, Mangalayatan University and the students and many people who are joined in the webinar. Everybody, welcome. And this is good morning from my side. Thank you for reading my CV. So today's my topic is I want to share now. I'll just share. Is it visible?
And just white background is visible, sir. Not at slides are not visible. White background is coming.
Okay.
Just open the slides. Open the slide show and just then share the screen, sir.
Yeah.
Now it is coming, sir. Now it
Yeah.
is coming.
Is it moving?
You can just move the slide. Move the slide, sir.
Yeah, moving. Is it moving?
It is not moving now. Only
It
is not moving.
I'm making moving. I'm moving now. Is it moving?
Not moving, sir.
Okay. Hey, why you stop?
I think first you share the slides and magnify the slides, and then you try to move it, sir. That will be better.
Put it this way. Now?
Magnify the slides, sir.
Yeah, we magnified.
Just move slide now.
Yeah, now?
It is not moving. It is not moving, sir.
Oh.
Some problem.
Wait a minute, sir.
Yes, sir. Otherwise, you send PPT to me also. I will also try.
Yeah. Hello, in close. Stop. Sharing. Sharing now. Huh? Sir, now is it coming?
Kishore, please come. It is not moving, sir. Coming slide is coming, but it is not moving.
No, I'm making now. Just to see now. Now?
No, it is not moving, sir.
No, it takes time. 1 minute. You see now?
Slide is not. His slide is not moving, sir. He is trying. Actually, slide is from shared from that side. It is not moving. No?
Okay, I'm sending to you, sir.
Now magnified, sir. It is magnified now.
Yeah.
Well, now try move, sir. Try move.
They have not shared the complete screen, sir.
Now you see, sir?
Now it is not moving, sir. Kishore, you tell, no? What is that you have to do?
Screen share lo complete screen తీసుకోవాలి అందుకని రావట్లేదు.
No, what he is telling is no complete screen you are not taken. He is telling.
In options.
In options, no, you are not taken complete screen. Otherwise, you send slides to me.
Yeah, I'm sending, sir. Do complete sharing control. Apologies. అంతగా మళ్ళీ షేర్ ఈమెయిల్ నేను పంపిస్తాను అప్పుడు ఈమెయిల్ దీంట్లో ఏం కానీ అంతగా పంపి ఫస్ట్ పంపించేసేది తర్వాత చేస్తాను సారీ Yahoo.
Can try that also. Sir, actually there are, when you share it, no, there will be three columns will come, sir. One is window, another is Chrome tab, and another is entire screen. You click on entire screen, sir.
Yes, sir.
And
Yes.
then select the slideshow.
Okay, sir. Okay.
And
Anyway, I'm
you
sending now slides.
Yes, sir. You send me, I will do that, sir. I will do it. No problem.
I've sent, sir, to your Gmail. You check it, your Gmail.
Email, huh? Okay.
Yeah.
Okay.
Gmail. There are three options. Entire screen, yeah. Share it. Now, is it moving?
Yes, sir.
It is moving.
Is it moving, sir?
I'm just checking, sir. It is move. Move it. Move, sir. It is moving. I think. You just move another slide.
Yeah.
It is not moving, sir.
Anyway, I've sent to you.
No, it is moving, sir. Sir, now move.
It is moving.
It is moving, sir. It is moving.
Okay. Okay.
It is coming, sir. Now.
Let it be. Yeah. Sorry for this thing.
No problem, sir. It is no problem.
Yeah. So today's my topic is biotechnology, how it is a promise to the farmers' prosperity, and what is the role in the next generation agriculture. This is a topic given by Honorable VC Samshwara Garu. So I will be talking about the biotechnological advances and how it is playing a role in a next generation agriculture. So before that, I just want to say about the status of agriculture across the globe, how it is. If you see 2023, across the globe, the small farmers produce 1/3 of world's food. The only small farmer who have less than 2.5 hectares. They are producing 1/3 of the world's food. And the small farmers are highly in China. And it is very, single digits in case of Brazil and Nigeria. That means Brazil and Nigeria are very highly, adapted and developed country in case of agriculture. As you all know, that corn, wheat, rice, and these are the world's most popular foods. And then if you see the agriculture per se, which is the country which is really contributing agriculture highest, is that's African country, that is Sierra Leone. That is contributing to the GDP of 60.45%, which is very high. I mean, this is the country which is highest GDP coming from the agriculture. And next is Niger. This is also African country. And then Ethiopia and like that. If you see in the Asia, Uzbekistan has the highest GDP from agriculture, like 23.47%. But if you see the top agriculture producers in the world, like China, India, or United States, or Brazil, they don't have much GDP contribution from the agriculture. That means their agriculture is not although they are producing a lot, but agriculture is not economical there. I mean, they are not contributing much to their economy. If come to the India is largest country, a producer of wheat, rice, and many commodities. It has either first rank or second rank, including dry fruits and many other roots, tubers, crops, pulses, and all, and numerous vegetables also. But growth rate of agriculture of India is only 3.6. It is a little bit was increased in the COVID time, but the growth rate, if you see average for the last 4-5 years, it is only CAGR of 3.6%. Why this agriculture contribution is to the GDP is low in the countries like India and many other top agricultural producers? If you see what are the issues of today's agriculture, the issues are almost common in across the globe. That is the worst. One issue is the climate change. That is a big issue. Soil erosion and biodiversity loss and changing consumer taste and expectations are increased a lot. It is, I mean, this is the one where people not earlier some taste they are preferring. Now they are asking for some other taste. This is clearly evident now in many cases. And growing demand for high quality food and high investment productivity. That means the labor costs and other costs are increasing a lot. Thereby, the productivity improvement for that there is you need to put a lot of money. So that is another problem. And maintaining a resilience in the face of global economic factors, because you have a crisis in some other country, then maintaining the resilience is also a big problem. And less percentage of youth coming to the agriculture is a big problem. Among all, I feel the big problem as a technocrat I see is the low adoption of new technologies. That I see as a big problem for the today's agriculture. And the technologies interventions, as in case of biotechnology interventions, there are several, but adoption rate is low. That's the reason agriculture is facing problem. Then I feel the biotechnology, what it can do, I'll tell next slide. But coming to the India's, how agriculture is important in India's perspective, everybody knows India is aiming for 5 trillion economy for by 2030 and 2047 to become a third best country. Then agricultural growth needs to be increased. Then in India, agriculture is a primary source for livelihood that everybody knows. But despite this GDP declining, but it still contributes significantly to the economy. Now in India, it is around 16 to 17%. It is not low compared to many other countries. So agriculture is important in India. Agriculture is also closer to rural development and poverty elevation, and ensuring food security for growing population is a crucial. And also India to become a potential to major player, focusing on quality improvement and strengthening agriculture value chains are very important. So that's the way India agriculture improvement is very well. But the problem is that facing lack of, I mean, a poor adoption of technology is a big problem in India also. So the technologies like biotechnology, I feel it is the potential which can transform the present agriculture to a next generation agriculture. This technology, I feel strongly that it can offer many innovative solutions by improving the efficiency, improving the productivity, and maintain the sustainability and bringing the resilience of agriculture systems. It can give the solution to all these four, which are the core for the agriculture. So biotechnology can offer. We can see one by one how it is doing. So what has been achieved so far? I mean, although biotechnology, we were talking from a long time, but in India or across the globe, what is biotechnology has done so far? That we can see. Then later on, we can see what is happening and what is still has to be happen. That we can see. So what is happened in that the most significant thing what I feel is the genetically modified crops. That is GM crops that has really given an impact in many countries, including India. And the crops have been now cultivating 32 crops are approved, and nearly 525 different transgenic events have been approved across the globe. And there are 71 countries adopted this GM crops and 24 developing countries. Among the 71, 24 developing countries are adopted this and grown to the percentage of 56 in the GM cropped area out of total cropped area. And the highest ones are USA, Brazil, Argentina. India has 11.6 million hectares. The global production of GM crops is rising. It is now almost like 112 fold increased compared to 96. So widely grown crops you may be knowing like soybean, maize, cotton. And in case of India, it is cotton. Cotton is the one which is highest, like 11 point, now it is 12.6 million hectares. Almost it is like 95% crop of 95 to, I can say it is now 98% of cotton is the transgenics we are growing. Now, there is no other than the transgenic we are not growing in India. So total in cotton, only transgenic crop is grown. So you know that in the transgenic there is a Bolgard Criven AC was the one which is approved commercially in 2002, followed by Bolgard 2. That was two genes, Criven AC and Criven 2AB, which is in 2006, which has a yield advantage of 245%. I mean, now if you see average productivity earlier, it was only 191 kg per hectare. Now it is increased to 469 kg per hectare. So that is the impact of biotechnology in India, especially by GM crops. So that is perceived, and many attempts are made in ICAR and universities and traditional universities, and then a lot of research is going on here now presently in different crops like cotton, rice, wheat, maize, brinjal, potato, and so on and so forth. And traits, especially the trait selected is herbicide tolerance, drought tolerance in initial stages, and later salinity tolerance, virus resistance were taken. But right now, most of the improvement in the GMO is happening in the quantitative traits, that is in the yield and nutritional improvement. That is now that's called as a third generation transgenics. So far now, 69 events entered into the regulatory system in India. Among these 69 events, around 22 events are from the public institutions. So that is what happening. I mean, a lot of things have happened, but now there was a moratorium for some of the traits, but hopefully this GM regulation, if it is make it clear in India, I think some more products may come, because there are already 69 events are entered into the regulatory systems. So this is what the impact from the GM crops. We can go to the second one, that is the molecular breeding. This is another tool, biotechnological tool, which came existence in the 1980, and now it is almost it is a complementing into the conventional breeding, and it has become a tool for all the classical breeder, because it is complementing greatly and it is enhancing the efficiency of the classical breeder to improve the traits very effectively using the molecular markers, because it can reliably and precisely characterizations can be done, and can be efficient and effective in terms of selection, and also in terms of time and also effort. So through molecular breeding, within a 3 season to 4 seasons, a cultivar can be developed. So there the traits can be integrated very effectively. There are different strategies are there in the molecular breeding, like marker-assisted selection, marker-assisted backcross broadcasting, or MABC or MABB, they call marker-assisted backcross breeding, and the new, I mean, relatively new, like marker-assisted recurrent selections and genome-wide selection and genomic selection. So these are all like a modifications in a same marker technology with a little bit of advance. When you go for recurrent selections, there the population can be improved effectively. And when you go to the genome-wide selection, there the markers will be selected entire genome by using a SNP markers. And genomic selection is using a SNP markers throughout the genome, and selecting a training population and going for the advanced population, where you can predict the genomic, you can give the values to the breeding. So that's called a genomic selection that is now happening in many of the crops. But what is the prerequisite for this molecular breeding is the most important, the markers and the genes and the maps, molecular maps. But some of the crops, genes are not known, even molecular maps are not known, then it is a little difficult. So that is the reason a lot of effort is going on in developing a linkage maps construction. Already 30 plant species have done, and work is underway to construct many more. And also sequencing efforts are going hugely in many crop plant genomes. Already India's contribution in the globe is 27 crop plant genome sequences were done. Maybe it is started from rice, now it is gone to pomegranate, several, including tea and many others. So 27 are India as contribution. Over the past, I think, globe, if you see, there are 1,000 genomes have been published, representing 788 species. That is the enormous work, progress is happening in the sequencing. You can see now almost 1,000 plant genomes have come and representing 788 species. And using this knowledge in ICR, several cultivars are developed. In rice only, if you see, there are 74 cultivars. No, it is not rice. It is altogether. If you see, there are 74 cultivars were developed, rice, wheat, and pulmit, chickpea, and soybean, and groundnut. So that is enormous products were released through molecular breeding. And because this doesn't have any regulations like, unless like earlier slide, like GMO, it is a classic, I mean, it is almost like bringing the genes from then you can take from a related species, or you can take from unadapted cultivars and can introduce into the cultivated one. So it is easy. So it is a lot of work is going on. If you see the next slide, you see there were many universities, many places across from the north to south. Many people are working on different crops like wheat, rice, maize in case of cereals. Pulses, there are many vegetables also, starting from chili to cucumber work is going on. Fruit crops also, enormous work is going on, guava and mango. Legumes, there are many, and even in spices like cumin and mustard. Medicinal aromatic plants also it is started. There are several universities, institutes are working, and enormous work is going across the India. Hopefully, I mean, this is the one technology which can develop the varieties very quickly and precisely. Whatever the trait you want, you can be integrated into the ruling variety. That's how this is. I mean, that's how this is revolutionary now for the agriculture problems. It's giving a very good solution either for, as I say, productivity or improving efficiency. So why this is so much? I just kept another slide explaining this. It's conventional breeding, as I said, that it takes 8 to 10 years because it involves hybridization, selection, backcrossing. I mean, there are techniques in conventional breeding like hybridization, selection, backcrossing, mutation breeding. All these ones, each one has its own advantages, of course, but the time taking is in this process of this all 4 techniques is at least 8 to 10 years. But whereas in molecular breeding, as I said, 1 to 2 year maximum, it takes 3 years. And there are 3 techniques which 3 types which I can say that marker-assisted backcross breeding, that is there involves like foreground selection, background selection, and recombinant selection. Whereas marker-assisted recurrent selection, this is by continuous crossing and selecting to increase the population and increase the favorable alleles in the population if you want to improve. And then this MARS can be used. Genomic selection, I said, it is upgraded from this marker selection where markers are selected on the whole genome coverage. So there are, I just kept 1 or 2 examples, which are mine only, which were the earlier examples where we used these markers and we cloned the genes from IRI with Dr. T.R. Sharma's group and Dr. Mohammed Hasan and Dr. N.K. Singh's group, where we identified MAC-based cloning was done for the rice blast resistant gene. Rice blast is caused by Magnum-Proteoglycia, is a fungal pathogen. And it is a very serious disease in rice. There were many genes were available by that time, but there were no genes which are effective in India. So we identified that one resource from the cultivar called T-tape. That was a Vietnamese cultivar, but it was showing high resistance. Then that cultivar was used and mapped the gene. You can see here, these are the markers, and we mapped here, and we fine mapped and cloned this gene, and and this gene is being used extensively. I think by this gene, by using this gene, already around 17 to 18 products were released in India and now still being used because it is offering resistance to the diverse isolates of blast from the north to south. That is the importance of this gene. And later on in IIRR, earlier it's called the DRR, Directorate of Rice Research, now it's called Indian Institute of Rice Research. I have cloned with my team that we cloned another gene called PA68T, which confers resistance for the both for the leaf blast and as well as neck blast. I think agriculture people know the neck blast is like it comes when the panicle, rice panicle comes out, then there is a incidence of this fungus in the base of the panicle, then the panicle get breaks. That's called neck blast. That is a severely it is coming because of climate change. And then PAKH was offering only partially to the neck blast, but highly to the leaf blast. Then we thought there is a need of identifying one more gene for the neck blast. Then we identified PA68, and that was isolated from not from any cultivated from, but it was isolated from Oreja glumipetula. This is another wild species of Oreja, and then we cloned and we saw this is showing very good resistance, and this also gene being used now in basmati and breeding and many other breeding programs, and now already one variety recently released and another 4 varieties are in the pipeline with this gene. So this is I just kept one example of web-based cloning and how these markers are used and how this mass is used in the development of cultivars. As I said, the mass to become successful, we need really a sequence of the genomes. And then the question comes that what kind of sequencing we need to do? Either really we need to put a lot of money, or what kind of sequence we need to do. There are, I mean, different methods you can choose. I just kept here. Long read technologies you can use. Even you want to use so for really, if you want to go for a good level of coverage. Then if you see there, the cost is more and the number of genomes are less. Only 100 are out of 2000, only 100 are there. Long read technology. But if you go to the exome sequencing, where you can go for developing only the markers by GBS, genotyping by sequencing, you can see large number of genomes already sequenced, because there you will get SNPs and alleles. So if you want to work in any of the species, you don't need to go for long read sequencing. We can go for exome sequencing or genotyping by sequence. That's called GBS. It is a random sequence which can generate a small 100 to 150 bases. That is enough to identify the SNPs. Thereby, you can use those information for SNPs identification. But if you want to go a little bit higher, then you have advantages. But for mass, I think I can consider GBS is enough. So once genes are known, once the sequence is known, we will have a question whether that gene is enough, or there are any different forms of that gene is available in the germplasm or in the whole genetic resource. That's the different forms of the gene is nothing but called alleles. The alleles can be identified for once gene is known, because diversity is enormous in India, especially in case of rice, because most of the examples I'm giving from the rice, because I worked for the 23 years in the rice. That's the reason I'm bringing a lot of examples from the rice. So a lot of diversity exists in the rice. Then we need to see whether we can have a better allele, or maybe we have a superior allele than what we have right now. That's a mining of those alleles called allele mining, because you have identified one allele from the T-tape, for that example, I can say, which is effective. But if is there any allele effective than the T-tape allele for the same gene, then we need to mine into the different land races or wild species. Then we can find out. So that's what we did for the rice blast resistant genes, some of the rice blast resistant genes, and we identified, of course, one allele from our own northeast land races, Amna Boha. I think Honorable VC knows this is from Tripura. Amna Boha is a land race which was really unadapted and doesn't have any yield. But the allele derived from Amna Boha was such a better resistance offered than the T-tape allele. So then we switch it to that allele now in integration into different background. So this is another PAB is another gene, and there are several genes we did. So that's what's the allele mining. That is another tool which can also aid in a crop improvement. So once we know the genes and once we know the alleles of that, but the function of those genes, how we determine, although you know the gene, but function, as I was saying, that it is offering resistance, how we know that that's the determination of the function is the work of called functional genomics. Now many of the genomes, as I said in the previous slides, that 1,000 to 2,000 genes are, 2,000 crop genomes are coming, then the more work is now is putting in the form of the functional genomics. The sequence information, which genes have which function. That is the now coming very well. That is the functional genomics. That can be done in a RNA level, that is called transcriptome. That can be done in a protein level, that is a proteome. And of course, going to the down is a metabolome in the metabolite, total metabolites of the genome. So this is the work is going many crops now. Functional genomic works is going on, especially transcriptome from the gene to what are the genes which are expressing at particular stage. That will be known from the transcriptome. Once we know that this gene is expressing at particular stage, is this a gene which is really imparting resistance or which imparting a nutrition, that can be known very well from the transcriptome. Once we know that this gene is imparting, then we can go to the what kind of protein it is conferring, and protein dynamics we can see, protein-protein interactions can know. Thereby, total gene function can be identified very comprehensively. This kind of functional genomics works now happening in many of the labs in India, across the globe also, because the structural genomics part almost completed in many of the cases. Now functional genomics is now going on very well. So in case of functional genomics, when we know that we have a different forms of the gene, when we have the different genes which are conferring either conferring resistance or conferring for nutrition or any other thing, then we can have a breeding strategy, a very new strategy which is just started maybe 3 years back, and in India it just starting now 1 or 2 years back only started. That's called haplotype breeding. Haplotype is nothing but a combination of alleles of different polymorphisms. If you see, this is germplasm. If you after sequencing, you will get like this. But there are some sequences which are going together. You can see AGT and here GTA and AGA. They are going together in the same chromosome. That is called haplotype. This is haplotype for a single gene. So suppose in a gene you have we have 4 genes. One may be disease resistance, one may be the nutrition, one may be the nutrient use efficiency. Another one may be a quality gene. Suppose we want to do, but because in the molecular breeding so far we are doing either 1 gene or maybe maximum 2 genes of the same trait. But when you want to develop multiple genes and multiple haplotypes, you want to put together, or even from it is derived from the different alleles, you have different haplotypes, then you want to put together, then this is the strategy. If you see gene 1, gene 2, gene 3, and you have a specific haplotype, gene 1, you have 4 H1 to H3, and in case of this, you have this thing, you have in case of this 4, then you want to put like H3, H2, and then H2, and this here H1, that is variety X. So like that, you can specifically put these haplotypes into the breeding, then you can make the designer one. So I want a, suppose example, I want a rice type which is having a low sugars, that is the best for, low glycemic index, that is governed by gene 4, and also I want a resistance which can for the blast, and also and short duration, and also it has a good phenotypic acceptability. All those traits can be put together and can design a kind of a crop. That's otherwise people call fashionably that designer crops is nothing but you're selecting a better alleles from the different parts and putting together. That's called haplotype breeding. A lot of work just started. Last 1-2 years. We also had a big project on this from the NASF, and the work is going on this. But just remember, it is not only the gene. it is the gene. Now we gone now gene to allele, from allele to haplotype. That means we are putting only blocks of haplotype of each gene and making the crop. So there is another techniques which is in a biotechnology, which is a new, relatively new, I can say, but many people know now. It is almost like a buzzword for everybody. That is called genomics, because why it has become so popular? Because it has tremendous applications in a agriculture, which can which I feel may coming days can revolutionize the agriculture. So what is genomics? It is making a specific change in the DNA of a cell or organism. Like that, in the previous slide, I said you are putting a haplotype consists of 3 nucleotides, but if you want to put only one nucleotide change, that you can do with genomics. 1 or 2 nucleotides, if you want to change, that can be done without doing much. I mean, it is simple. Then the what haplotype breeding is there. So it is used to correct, introduce, delete almost any DNA sequence in many different types of cells and organisms at one time if you do, and it can percolate to all the cells and all the tissues. So it is done mostly using enzymes, particularly nucleases, that have been engineered to target specific DNA sequence, where introduce and cut the DNA strands, enabling removing and existing DNA and insertion or replacement. That can be done. Although there are several tools before CRISPR, like JDFN, ring finger nuclease, TALENs, CRISPR, meganuclease. But CRISPR, I mean, no, man, I think even students know now that CRISPR-Cas is really become a popular because it is faster, cheaper, and more accurate than other methods. There are some disadvantages which I am really not going to much into the details, but you can see that they have some disadvantage of JDFNs and TALENs. That is the reason the CRISPR has become more popular, because it has all the advantages of editing. So I just to tell one more slide on the genome editing, how it works. I mean, simply, I just want to say that this is not a new thing. Actually, genome editing is exist in a bacteria. To invade the protect from the virus. So that was there available. So that is the one mechanism was existing, that was using now with this editing technology, and now using for the modification. How it works, if you see, virus invades in the bacterial cell, the DNA molecule enter into there, that is transformed into the RNA, and it will integrate here, and it will become a CRISPR RNA will form. This CRISPR RNA will guide the nucleases to cut the precisely. So that's what's happening. So now artificially, when you want to change any DNA or any of the modification, we need to develop this sgRNAs. sgRNAs you need to design, and you need to put this sgRNA related to where you want to intended to change, and then you want to introduce into the plant. Then with the CRISP nuclease will be there in the vector that will cut and make the changes. So how it is happening? There are 2-3 types are there, like side-directed nuclease 1, side-directed nuclease 2, side-directed nuclease 3. This is now called SDN1, SDN2. This is an acronym everybody using now. I think we should know that there are 3 categories are there. In SDN1, it changes small insertions. It is only 1 or 2 nucleotides it will be changed. You can see here 1 or 2 nucleotides you can change without any foreign template. That is SDN1. Whereas in SDN2, it is uses a small template, but doesn't change much. It changes also 1 or 2 changes, or maybe it deletes 1 or 2. Excuse me. So SDN2 also is a only small change. But whereas SDN3, it needs a template, full length of gene, either maybe a foreign or maybe the cis. So that SDN is requires a complete length of foreign or maybe the cis. So that is SDN3. That is the reason now in India recently SDN1 and SDN2 is deregulated. That means that doesn't require the regulation. because it doesn't have any foreign material. Whereas SDN3 is like a GMO, like what I said is like earlier transgenics, so it is not deregulated. Whereas SDN1 and SDN2 is deregulated now in India, and it is many more products are coming now. In rice, at least two products will come in next year. Now it is already in the final year of testing. Similarly, many labs, including my lab in tobacco, we are doing genome editing, and hopefully we can produce. I will tell that what we are doing. If we see the current status of genome editing throughout the world, so first one came in Japan, that is tomato, high GABA tomatoes, which increase gamma aminobutyric acid that to reduce the blood pressure and promote the relaxation. This is a product came in 2021. I mean, almost 3 years back. And in second one came in maize, high starchy maize. Again in the Japan, it is amylopectin increases, and thereby starch is increased. And India, the India status is that SDN1 and SDN2, as I said, into last year, is the DBT, Department of Biotechnology, announced that it can be a deregulated. That's the reason I kept as a green. You can see here it is a green. Most of the places you can see it is SDN1 and SDN2. It is a green now, except to some places in a case of Europe. But otherwise, it is in a throughout world. It is genome edited products are deregulated, doesn't come under regulation. So there is a tremendous scope for this technology to offer the solution for the current problems. That's how you see the market of the genome editing compound annual growth rate is now 15%. I think this slide is a little low. I mean, but it may increase even more than 50%. If you see 2022, it was 4.6. Now it is jumped. It is jumping now. I mean, it is an annual growth rate of around 15%. So that is this thing. But I also would like to bring you that whatever the change we do, either you do the change in the gene, or you change in a one gene, or you change in a two genes, or you change a allele for that, or you use a haplotype, but that has to be done in a whole systematic approach. I mean, it should have a total interaction need to be taken into account. That is called system biology. System biology is nothing but is a holistic approach. It is full understanding of the biological system. That will combine the whole genome, transcriptome, epigenome, proteomic, and all, and also together with the modeling and high-performance computation analysis. Then only whole systems approach can happen. Because sometimes what happens, you are changing one gene for salinity, but it has other effects, and also it is not making a holistically, our system is not system is not efficiently increasing. So that is the reason the system biology approach has come now. So that is integrate and interact with the network of genes, proteins, biochemical reactions, and from various internal and external environment. So they do first a model with whether if you change this thing, what will happen. They will create a model and do the experiment and analysis. Then they do the kind of changes. That is called a system biology approach. And now being, I think this system biology approach now being followed in a wheat and barley in cases like cement and other, I think this is not came still into the, not in a practice right now, but I think in coming days, the system biology will definitely, because, data analytics, data is coming in a large number of data is coming in a different places, different environments, and different crops. So whether if you change this one with this environment, what will happen, whether really it will solve or not. So that model can be created and can we do the experiment and then can really see the effect of that. That is the system biology approach that is coming now. And of course, not only so far I said maybe most of the time I said about crop improvement, but the crop improvement not in the breeding per se. You can also, biotechnology offers solutions for the pest management, because the pests like insects or diseases, there are tremendous, they will reduce the create a lot of loss. How the biotechnology is offering? The first one is detection methods. Many of the times the accurate detection of the rays or accurate detection of the pathotype is very important. It was done mostly earlier in phenotyping and other methods, but now DNA-based detection is very specific, and it can be done either through fluorescent in situ ablation or spectral karyotype imaging or DNA chip technology, which we can identify particularly very effectively that this is the race which is infecting blast or either blast or any other disease. That's why we could identify very nicely in case of wheat rust and many other, and then the detection is now that is offered by biotechnological tools. And there are other method like RNA interference. This is another tool, biotechnological tool. I say it is really effective in controlling the insect space. So because this is also involves sequence-specific gene silencing, we mostly use a gene silencing method using a double-stranded RNA. You can see here, this is the siRNAs, and risk complex will form, and then it will make the silence. Mostly this is used for silencing the pest genes, either pest genes, importantly insects or diseases, wherever the pests are important genes, suppose like some genes which are important for digestion, some are important for development, that can be silenced easily by using this dsRNAs. This DNA, double-stranded RNAs can be synthesized chemically and also can be sprayed into the plant, or double-stranded RNAs can be introduced into the plant and can create the you cannot call it transgenic, but you can create the event in that. So that is possible. And with that, at least in my lab at ICAR, we could silence two genes like cytochrome P6 and APN. These two genes were important were for YSB, rice yellow stem borer, plus we silenced these two genes through dsRNAs introduced into the plants. So that's how, there are many. In case of forest trees, in case of trees like sandalwood and all, now in forestry, for controlling the pest, dsRNAs are spraying. But only thing, the stabilization is a little problem of these dsRNAs when the environment is harsh environment. But now the technology come with the stabilizers. I think this dsRNAs are playing a better role in controlling the pests, especially insects and diseases. There are several examples I kept here. I don't want to read, but it is there. RNAi successfully used in many crops, starting from tobacco, now peas and wheat and crucifer crops and other crops. And also not only crops, but even pest genomes are getting sequenced now. Many. If you see the pests, there are around 20 insect pests, including rice stem borer, rice yellow stem borer, we sequenced at ICAR in my lab. We sequenced the first time we, internationally we are the first one. We sequenced the yellow stem borer completely, and rough genome was published. And like that, there are several groups are publishing several insect species, important pests like you can see here aphids, and then striped rice stem borer, and mustard beetle. There are really Spodoptera. There are very important pests. So once we know pest genome, when you can know how these interactions, host pest interaction or host pathogen interaction. Similarly, many diseases also. Many disease causing blast was sequenced long back. This Magnoparthae, but many other disease causing organisms got sequenced. With that, we can know what are the susceptibility factors, what are the ones which are making this host as susceptible. When we can make them resistant easily. So host pest interactions understanding, I think the genome sequence of the pests are really helping. And the most importantly, now the effectors are the proteins that secretes into the plants. That's called effectors. That is now understanding coming very largely. Then that helping to develop the resistant cultivars or resistant mechanisms, helping lot. So not only our crops, but even pest genomes coming a very huge number. So biotechnology also offering for the food quality and nutrition, which I was telling from the beginning, not only resistance and all, but we also enrich. You can see here, this is a golden rice, you may be knowing, enriched with a beta carotene, vitamin A, and now enriched with the proteins and essential nutrients, zinc, iron. There are many more. Zinc, iron were done mostly in the molecular breeding methods, but in case of iron, it is done with the case of transgenics. And also not only increasing in nutrition and also decreasing the spoilage, reducing the spoilage. That is also one important. And reducing the natural food toxins like aflatoxin or other toxins are there in some cases. That reducing also we can do with the biotechnology. And also biotechnology is helping to deliver system, medicines, foods as a deliver, foods as a medicine now. Food as a medicine because it has all the medical compound can be kept here and then give as a food as a medicine. That is also happening because it is easy to deliver and it reduces spoilage, no refrigeration required. Like in banana was used many cases as a food as a medicine because banana was tissue culture and transformation is very easy. So it was done and it is being done in some cases. So food is used as a medicine. So, and also, when I say the nutritional security, biotechnology tools were used for bringing a nutritional security. Earlier, we had a target of giving a security of the food, but now the age giving for the nutrition. What we are eating is not important, but what the nutrition we are getting, that is more important. For that, of course, all the crops it is started, all the methods like genomics, transcriptome, proteome, metabolome, many other methods, all the methods which I told earlier were used or integrated into becoming the nutritional security to develop either functional food or to develop a better better products. Novel method products are coming for the nutritional security. And not only that, biotechnology in the agriculture, if you say agriculture, the ecosystem restoration is very important because unless ecosystem, because what I say, genotype into environment interactions are very important. So ecosystem restoration also now biotechnology is being very well used. I mean, not only this drought tolerant crops, nitrogen use efficient crops, nutrient use efficient crops are coming usually, and climate smart traits identification and genes is happening. Bioremediation, this is the one where I feel the restoration is mostly in that context, I kept here bioremediation using living organisms like microbe, bacteria for the removal of the contaminants, pollutants, toxins is being done, and there biotechnology is playing very big role. So in ecosystem restoration, of course, biofuels. Now biofuels are coming from many plant materials like corn stalks, grass, many way because biofuels have a very good environment. Green energy that is now is being used biodiesels and all. There are a lot of progress happened, at least in case of corn and sorghum biofuels, and even from the sugarcane is coming. Tremendous progress has happened. Of course, biofertilizers, biopesticides, microbial biopesticides, these are all being used. Although there are many more, many microbial biopesticides are being used in agriculture, which sorry, which are offering solutions. And as I said in the first slide, they increase the efficiency and also increase the total efficiency of agriculture. These biopesticide and biofertilizers are being done, but lot have to done in a lot has to be done, like selection of effective strain and also production of inoculants and preparation of carriers. Of course, this is a continuous process still being happening. But I just want to flash for the students' sake, there is a one thing it is now is a DSI, digital sequence information, that is the data stored digitally, such as DNA or RNA, that describes organism characteristics and the trait. Because the DSIs we need to develop now, otherwise, for resources, all the genetic resources, GSS development is now very much required. And when you want to share this information, you need to give the DSI information. That's how that is taken a policy in many of the CGI states. DSI is developing now in India. So recently, the NASA has made a brainstorming session on this, and DSI is important for the biodiversity conservation. The last slide, what I want to say, I want to summarize what I said is how whatever the agriculture, what we are happening when I entered into the BSc Agriculture in the 1990, agriculture was like a subsistence farming. It is only to give a sustenance, maybe for livelihood. But now the agriculture is now getting transformed into the commercial agriculture. So how it can be transformed very quickly, I can say that the selection in the breeding process, the selection should be on the genotype, not on the phenotype, because we have the genes are known. So that is the reason we need to do the selection on the genotype and exploitation of the genes. Not that, I mean, you know the trait, but you need to know, exploit the genes for the breeding, not just by trait selection, but to use the genes for the trait selection. That is exploitation of the genes has to be done, and incorporation of climate smart traits. Let it be a heat tolerance, let it be a cold tolerance, let it be a drought tolerance, let it be a waterlogging, let it be anything. The genes are there, though. So we have to, I mean, the more emphasis need to be done on climate smart traits and breeding for mechanization. Many times I said in the one of the biggest problem, I said that production cost is increasing. How to reduce the production cost? We need to have a mechanization. But many of the times when you do the mechanization, our crop plants not amenable to for the mechanization. So sometimes we need to change the architecture of the plant type. So we need to do the breeding even for the amenable for a mechanization. So this is what now not happening, but slowly people are started to doing that. And breeding for consumer demand. So consumers are really asking very differently. An example I just kept here, you can see in the tobacco consumers, they want to consume the tobacco, but they want to make it less harmful. Can you make it a less harmful tobacco? That's what the consumer is asking. I mean, although a little bit reduced the tobacco consumers, but whoever are consuming, they're asking that less harmful tobacco. Like that in the case of rice, they're eating the rice, but they want a low starch rice. That is a low GI. So that like that, the consumer demand is changing. For that, you need to do. With this less harmful tobacco, we are using genome editing to reduce one of the biggest carcinogenic compound called tobacco-specific nitrosamines. These are the group of compound we are using a genome editing to make this less harmful. I cannot say it is safe tobacco, but I can say that is less harmful. At least in case of carcinogenicity can be reduced. Similarly, low lignin sporoustis in case of tree. And other thing is that microbiome, harnessing microbiome. I mean, in the microbiome, what are the things are there? That harnessing for the productivity is one of the another factor we need to think seriously for the productivity and focus on the quality improvement and the value addition. This is the work we are right now doing, value addition and quality improvement, using of artificial intelligence and big data for decision making, integration of biotechnology with other technologies like precision farming definitely makes a commercial agriculture as a viable. These are the 5Gs which Rajiv Vashne says that we need to harness into 5Gs like genome, germplasm, genes, genome breeding, and genome editing. That's why the agriculture can be increased tremendously. That is the reason the institute of mine is now changed into the new name called NIRCA, National Institute for Research and Commercial Agriculture. So this is one of the unique institute now. We are working on 5 crops apart from the when tobacco is there, but we are also working on turmeric, chilies, ashwagandha, and castor. What we are working on genetic enhancement of commercial crops for doubling the export potential. We are not doing an yield enhancement, but only an export promotions. That means more of value addition and post harvesting, and also development of energy efficient technologies for sustainable production and enhancing the commercial value of crops through novel post harvest produce management and value addition approaches, and exploring the innovative extension methods and using the market intelligence approaches for efficient market and profit realization. Because most of the times you produce, but where to sell, that is an important thing. So we are also using market intelligence approaches where the international and domestic markets are there, where we can export, what are the destiny countries for these products. That's what we are doing. So to our target to achieve sustainable commercial agriculture, that's what our institute is working. So hopefully in coming days we can really with using technologies like biotechnology and other technologies, we will bring a happiness among the farmers. This is our institute logo, National Institute for Research and Commercial Agriculture. We are working right now on 5 crops, but in a future we may include some more crops too in commercial agriculture. So in the lastly, what I can say that on biotechnology coupled with all the technologies can really can make this agriculture and present agriculture situation to the commercial agriculture. That's how next generation agriculture will be there. So thank you all. Thank you for your patience hearing. Thank you.
Thanks, sir. Thank you very much. I'm very happy that I have to inform everyone that even though you are traveling since morning 4 o'clock, you took a lot of pains in coming and taking this lecture to our participants. I'm really thankful for that because I know that you are traveling since morning 4 o'clock. Over to E.T. Madam.
Thank you so much, sir. Thank you, sir, for your presentation. Now the forum is open for questions. If anybody is having any questions, they can have interaction with sir.
They can write in message, why if they want, they can write message in chat box, or otherwise if anyone has a specific they can interact. Students, they can interact.
Students, do you have any questions? I think they are not having any questions because it was a molecular biology topic and it is really very heavy for our students who are studying in bachelor's right now. So if they are having any questions, we will write to you, sir, for the same. Now I would request Dr. Priyanka Dubey to vote of thanks.
There is one question actually. Sheshu Madhav, there is a message, how we can tell us about molecular biology to farmers?
Yeah, we no need to tell about the molecular biology to the farmers, but the products coming out from the molecular biology, you can tell that this is the one which came from this, and that has a effect. That has effect on the agriculture, and that has a advantage over the other conventional breeding methods. So that's how you can tell. No need to tell the process, but you can tell the products, whatever is coming, which has the offering solution, so that you can tell to the farmers.
Molecular biology is not required for farmers, but their application part is required for farmers actually.
Yeah, yes, sir. So farmers are Indian farmers are very knowledgeable. Don't think that way. If any product has advantage, any product has a better than what they have, they will jump on that. One example I'll tell you, the transgenic cotton. In India now, we don't have any cotton other than the transgenic. So that's the beauty. So because it is very good, so it is immediately picked up. Yes, sir.
So, anyone, any more? Is molecular biology product is safe for consuming?
Yeah, that is a question. It's a good question. I can say molecular products, which I told that, and among the molecular products, like the products coming from the molecular breeding, is very safe. They are nothing but anything, nothing but the conventional varieties. Even the products coming from genome editing are safe. We are seeing that even the products of the GMO transgenics, which they are so far we don't have any report on non-safety in case of tobacco. But when you coming to the GMOs in case of food, but any product which is coming to the if it is released, that will undergo all the tests. They undergo several tests. Then only it will be released. That's the reason now in India there were people who are having some safety issues, but it was several tests has done. Then only it is released. So once it releases, you can say that it is safe.
Okay. Okay. Over.
I think Mr. Abhishek, your question is clear now. So now I would invite Dr. Priyanka Mishra Dubey for extending the vote of thanks.
Good afternoon, everyone. I, Dr. Priyanka Mishra, on behalf of Faculty of Agriculture, wanted to take a moment to express my heartfelt gratitude for the wonderful opportunity provided by our Honorable Vice Chancellor Sir for organizing the webinar on biotechnology, a promise to farmers' prosperity and next generation agriculture. I am also thankful to Dr. M.S. Madhav Sir for providing his valuable time and sharing useful information with our students and participants. I also thankful to respected Prof. Madam, Registrar Sir, Dean Sir, respected faculty members, participants, and my dear students. I would like to convey my thanks to Mr. Pavan and Dr. Satsangi for providing technical support. I hope this webinar will definitely be useful for the students and for the participants. Thank you, sir.
Thank you.
Thank you, sir. Thank you very much.
Thank you all. Yeah, thank you all for joining. And if you have any questions or anything, you can write to us. Definitely for the biotechnology on any aspect, we will answer. There was how about addressing the pest developing resistance to new modified crops? Yeah, you need not to worry about the resistance development now. That's the reason what we are saying is we are not putting a resistant genes. What we are doing is we are silencing the genes which are important for the pests. So that's how.
There is one question came just now. If you are able to please see, sir. How about addressing the pests developing resistance to new modified crops?
Yeah, yes, sir. That's what I'm answering. That you no need to worry that whether pests will develop a resistance. The strategies that using resistant genes is not the approach now. What we are doing is we are modifying the susceptible genes which are there in the plant. So that because susceptible genes are there in the plant, which helping the pathogen or pest to come. So now when you modify these susceptible genes, then there is no chance of interaction. So development of resistance will become very slow, very slow. I don't say that it will not develop, but it will take long time. That is the one approach. The second thing is the genes which are important for the pests to infect into the host, that also getting now silenced. So now the even for the pests, it is becoming ineffective. So that's the way the resistant development is getting delayed. So that is the new methods of development of pest resistant crops. So definitely this will offer more resistance for the long time. Yes, sir.
There is one more question. They are not leaving you. Sorry, if transgenic plant is important, then why government not show any approach, and why BT brinjal, BT okra is banned in India?
Yeah, this is again a good question. This is again a good question. Yes, as a students or as a faculty, you should know that transgenics, when you talk as a transgenics, when you're putting either a foreign gene or maybe related gene we are going from other system, when undergo the several tests with the unmodified one, which is similar to that, and having a advantage over either nutrition or maybe in case of BT brinjal, he said BT okra, it is having advantage over other one having a resistance for the insects. When it is there, it can be released, but that is the reason this kind of webinars or this kind of interaction to the students is really helpful, because the mindset we need to change. I mean, we need to think that it not like general people. We should not think that it is a non-safety or it has something. It doesn't, because it has shown in many. I mean, I said that there are around 57 countries. BT brinjal is approved in Bangladesh, but there were no report that so far anybody died because of this eating BT brinjal. This is only a myth. I mean, one should have a kind of a, what I can say, kind of a suspect, but one cannot go much beyond to that. There are several evidences saying that it doesn't interact, it doesn't have any target in the human, it doesn't, protein will not be there. So many things are there. Still, people think that it is not. So that is the reason policy makers and some of the NGOs are making hue and cry and not making to release some of the products in India. But as a students of agriculture, as a faculty of agriculture, you must know what is real, what is not unreal. Then I think we can progress more. Thank you.
Thank you, sir. Thank you very much. Okay, sir. Thank you, sir. Now you can end the session. Yes, sir. Thank you, sir.
Yeah, thank you very much. I think the good questions, and so Professor Rao, SK sir has
SK sir is there.
Yes.
Yes, sir.
Thank you very much,
Thank you, sir.
Dr. Sheshu Madhav. You have given excellent information. I know your credentials and all these things for the
Sir.
last many years in the biotechnology area.
Sir.
And I think really informative lecture for the students and as well as the faculty members. Thank you very much. We will meet once.
Thank you, sir. Thank you very much, sir. Thank you, sir. Thank
Thank
you,
you,
sir.
sir.
Thank you,
Thank you,
sir.
sir.
Thank you, sir.